The mouse incisor is remarkable in its ability to grow continuously throughout the lifespan of the animal. As a mouse gnaws on food, the tip of the incisor is gradually worn off. The constant abrasion of the tooth is counterbalanced by the production of new cells at a regular rate from stem cells. This allows the length of the incisor to be maintained and its function in feeding and survival of the animal to be ensured. Embedded in the jawbone, at the proximal end of the incisor, are two types of stem cell populations. Mesenchymal stem cells populate the dental pulp and give rise to the odontoblasts, a specialized cell type for dentin secretion, while epithelial stem cells produce enamel-secreting ameloblasts. Together, they generate all the necessary differentiated cell types in order to lay down new minerals and maintain tissue homeostasis. The structure that houses all the epithelial stem cells for ameloblast production is called the labial cervical loop. A similar, but much smaller, structure can also be found on the side facing the tongue. It's called the lingual cervical loop, although its function is less understood. Within the labial cervical loop, epithelial stem cells reside in the outer part of the enamel epithelium. They express several stem cell markers, such as GLI1, BMI1, and SOX2. To produce new cells, epithelial stem cells first give rise to a group of highly proliferative cells known as transit amplifying cells in the inner part of the enamel epithelium. These cells undergo a limited number of cell cycles and divide perpendicularly relative to the basement membrane. As new cells are being produced, older cells move further distally along the length of the incisor and begin to differentiate. Differentiating ameloblasts in the mouse incisor move at a rate of approximately 180 micron in a single day. Eventually, the cells differentiate into mature ameloblasts and start to deposit the enamel matrix. Many of the ameloblasts undergo apoptosis during this process, while the remaining cells shrink in size and regulate enamel maturation. Dental mesenchyme is quite heterogeneous and displays great homeostatic and regenerative capacities. The lineage hierarchy in this complex system is an area of increasing interest, about which we currently know relatively little.